In this video, we focus on writing a method section for a chemistry journal paper. Please make sure that you have opened your Google Doc and you're ready to write comments in order to reply to any questions posed during the session. We have already talked about IMRD papers and the sections they normally include. You can see them on the left-hand side. In fact, the method section is the section that many chemists write first. In part because this section describes what they know best, the procedures they have repeated many times to conduct their work. Interestingly, once they have established the select methods, most research groups use similar methodologies for several years. Hence, previously written method sections often serve as models for writing new method section. Together, these factors make the method section one of the easier sections to write and an excellent place to begin your writing journey. By the end of this chapter, you will be able to do the following. Know how to address the method section audience from a variety of chemistry fields. You will know how to recognize which details to include and exclude from a method section, depending on the journal you write for. You will also be able to organize the method section following a standard layout or moves. You will know how to use conventions such as capitalization, abbreviations, numbers and units appropriately and correctly. And you will know how to, hopefully you'll know how to use academic style in a conventional manner. The purpose of the method section is to address how a particular work was conducted. Relevant information about instrumentation and experimental or numerical procedures is described. The goal is to describe the information in enough detail that an expert, not a novice, could repeat the work. This is to say that in a journal, certain details are included and certain details are expected to be omitted. Let's write our first task. Are you ready? There is a variety of names normally corresponding to this section. Can you write down what you can think of as a heading or another title instead of just methods? What other titles or headings are there? You can pause the video here and write your answer in your Google Doc. There you go. Usually this section is formally called, for example, materials and methods or experimental section, but for brevity we call it simply the method section. Mind you, you're not completely free as to what you call it, as in each journal they tend to go for either one or the other heading. Are method sections in a journal paper similar to lab reports method section? Because method sections of journal articles are written mainly for experts. They're not at all like method sections of chemistry lab reports. Lab reports are written largely for instructional purposes, to reinforce new techniques and help students carry out experiments successfully. And as such, lab reports tend to include details such as lists of equipment, safety precautions, and step-by-step directions not needed or wanted by expert readers. On the other hand, experts expect you to leave out such details and magically make your writing more concise. In fact, concise writing is very important in journals because, unlike a single lab experiment, method sections describe multifaceted works that might have taken months or years to complete. Similarly, lab reports often include language inappropriate for journal articles. For example, the method sections of a lab report, a student might write stir mixture, heat to reflux. In a journal article, however, this would be restated using past tense and passive voice, as in the mixture was stirred and heated to reflux, making the writing more formal. We will talk about this again later on in more detail, but now it's time for you to write what other purposes does the method section in a journal paper serve. Pause the video and take your time to write down your thoughts. Well done. The journal article is not intended to teach a novice chemist how to conduct a first experiment. It serves as a blueprint for the experienced chemist. The recipe is included, but cooking instructions are not. The major purpose of a method section is to describe for other chemists, the audience, the procedures that were used to obtain the results presented in the article. A well-written method section serves as a resource for expert chemists who wish to develop similar procedures, compare their own procedures with those presented, or familiarize themselves with procedures in a branch of chemistry other than their own. As such, it determines a study's validity and it ensures transparency and reproducibility of the work. Many authors report that they begin writing this section while conducting the experiments. Yet, before you start writing, you need to make sure you have found adequate sources to use as citations. Once you have all the data, you can prepare your schemes, your figures, and your tables, and match the order of this section, the method section, to the order of the results section. At this point, write down when you think is best to write the method section. Let's have a look at this slide. It is often reported that the number of citations in the method section is very low or even negligible. However, this is not always the case. Look at the following sentences, which are taken from published papers, and identify the reason why they have used the citation in this section. The first one is a self-citation. Obviously, these researchers are publishing following a similar research path they had previously published. Regarding the third one, there are molecular dynamic simulation tools used with NMR that allow multiple topology and parameter files to be used simultaneously, but many chemists use pre-combined files. Now, in your Google Doc, write why you think the second and the last citations are used in methods. You can pause the video now. You don't need to rush. To get a better idea of what can or cannot be included in this section, we need to make sure we comply with the author guidelines of the journal we intend to publish to. Mind you, there are loads of journals and each one of them is different even slightly. Generally speaking, the author guidelines describe the details they want in your method section, the format, the citation, and the differences in your work when comparing with others cited. Here we can see an example coming from the Journal of Environmental Science and Technology, and you can see they talk about the length, they talk about what they focus on, they talk about formatting, the SI unit, and they say that the details can be placed in the SI unit. The SI stands for Supplementary Information, which is online only. It's a peer-reviewed material that is essential background to the article. I mean, it provides large data sets, methods, calculations, but which is too large or impractical or of interest only to a few specialists to justify inclusion in the printed version of the paper. In Nature, in the journal Nature, for example, supplementary information is not supposed to contain figures. Tables may be included, but only if they are unsuitable due to containing large data sets. In this video, we're not going to talk extensively about supplementary information, but I guess this is something you should do when you're with your supervisor the first time you write a paper. Let's have a look at the author guidelines in inorganic chemistry journal, in an inorganic chemistry journal. For example, here, as you can see, there is a considerable amount of specificity in a journal like this. For example, fully characterized compounds should have bolded compound names and structure numbers as the titles of the paragraphs in which their preparation, isolation, purification, and properties are described. When it comes to intermediates in multi-step sequences that have not been purified and fully characterized should not have their names bolded. Their preparation and partial characterization should be described as a step in the synthesis of a fully characterized bold titled compound. Reactant, reagent, and catalyst quantities should be given in both weight and molar units. Reaction solvent volumes and reaction times should also be reported. Use of standard abbreviations or unambiguous molecular formulas for reagents and solvents and of structure numbers rather than chemical names are used to identify starting materials and intermediates. All reported yields should represent weighed amounts of isolated and purified products and must be reported in the experimental section as both weights and percentages. When a series of related compounds has been prepared using substantially the same procedure, it is usually sufficient to present a single representative example. Examples can include, along with the yields, the weights and molar amounts of the limiting reactants, as you can see here. So it's very detailed and it varies from journal to journal. Now, let's have a look at more conventions by noticing what and how many conventions this excerpt adheres to. So this is an excerpt from the Journal of Organic Chemistry. I think it's issue 22. I don't remember the author at the moment. At this point, what I would like you to do is to read it and write down what conventions I just mentioned. You can also identify in this one. You don't have to go back to the video, rewind the video and find the table. You can also find the table in your Google Doc. You can pause the video now. Great work so far. Let's move on. In the beginning, we said that you will learn how to organize your method section following a standard layout or moves. In order to do so, you need to notice these moves or information elements yourself. But before you do that, I would like you to write down the layout based on your current experience as a student. You do not need to read or see anything yet. Just write the layout as you believe it should be included. It should be written including a relative degree of detail. You can pause the video now if you like. Most method sections follow a conventional organizational pattern. This pattern typically involves 2 or 3 separate steps, each of which corresponds to a move. Now it is time to go to your Google Doc and check if you have made an estimated guess regarding the layout of the method section. So, read excerpt A and make changes to your initial layout if necessary. You can pause the video here. Great so far. Let's have a look at the general chemistry method layout. As you can see, we have 3 moves. 1, 2, 3, all in bolded letters. So move 1 tells readers about the materials used in the work. Materials refers to chemicals such as solvents and reagents. It may refer to samples such as soil, water, or food, or it might include biological media. For example, bacterial cell cultures and other items used to conduct the work, such as the solid phase microextraction fiber, the SPME fiber, used in the excerpt you read. In field studies, the sampling site is also described in this move. When different types of materials are used in one work, subheadings, shown here in bolded font, are commonly used to set them off from one another. You can see the subheadings in Excerpt A, the subheadings chemicals, biosamples, SPME fibers were used. Subheadings actually are commonly used in Move 1 and they include the following: reagents and materials, samples, cell cultures, and site description. You can see the other 2 moves further down, like describe experimental methods and describe numerical methods, for which we're going to talk about in the next few slides. But before we do that, let's have a look at the level of detail and formality that is required to adhere to when we write Move 1. We need to report the name, the purity, and the vendor for all essential chemicals and materials used. We also need to know that when more than one vendor is involved, chemicals are typically grouped by vendor. And last but not least, the location is reported at the end of the sentence in brackets. Now, you need to identify a sentence in Excerpt A that actually mentions vendors and location. Can you see it? Where is it? When you find it, write a comment in your Google Doc. Now let's have a look at the sentences and write the words correct or incorrect to describe how the conventions are used here. You can pause the video here to write the answers, or you can just do one at a time. So let's have a look at the first one. The second one. And the third one. Pause the video to write your answers. So at this point, you have seen move 1, describe materials in the method section of a chemistry paper. Now I want you to read excerpt B again and tell me if what we just said about move 1 applies in inorganic synthesis papers. What appears to be move 1 for organic synthesis? Read the excerpt and pause the video to write down your answer. As we can see, move 1 in excerpt B is quite different. And in organic synthesis, they start with a brief description of the instrument and its operating conditions. They do not describe all the chemicals used in the beginning, but much later on, when individual syntheses are described. Samples are treated much like chemicals. If sample collection follows an established procedure, that should be noted in the text. So as we see, move 1 is approached slightly differently in synthesis papers. Such papers typically describe a series of related reactions, often totaling 10 or more individual syntheses. Rather than describe all of the chemicals used for these many reactions at the start of the method section, authors instead only use general information in move 1. And a very common subheading for this move is general. Now, let's see what goes after move 1 in excerpt A, or in other words, what is the next information element? You can pause the video here. The second move in excerpt A, and a common chemistry paper, a typical chemistry paper, is to describe experimental methods. The authors describe how they obtained their data. The move involves 2 sub-moves. The first sub-move describes procedures, includes analytical procedures, the steps used to prepare, to extract, to concentrate, or to derivatize a sample, field collection procedures, the steps used to collect water samples from a polluted lake, for example, and also synthetic procedures, the steps used to synthesize target compounds, to name only but a few. In some journals, particularly those describing analytical procedures, this sub-move also includes procedures used to ensure the accuracy and precision of the work. Such procedures are described as quality assurance and quality control, and you can see an abbreviation here. This abbreviation refers to quality assurance and quality control, and it involves a standardized method used to verify the quality of the data. Subheadings are also common in move 2. As in move 1, subheadings help organize a paper that uses multiple methods. But let's say a few more things about quality assurance and quality control. Sometimes this is embedded in the methods, so it doesn't have a subheading, but sometimes it is a separate section with a subheading. Let's have a look at an example. As you can see, this section is not embedded, but is a separate section with subheadings. When you write about quality assurance and control, it is important to realize that we are expected to use a certain number of terms. Let's see whether you can identify the key terms in these sections. Please pause the video and take your time to write your answers. You can choose words you even suspect are correct, even if you're not really sure of what they mean, as I will explain them soon after. There you go. You can see what words are highlighted. You're expected to use words like matrix spikes, surrogates, recovery, internal standards, accuracy, and so on. Blanks. These terms have been have been defined in the table below. You can see what spikes are and the rest of the terms. But let's have a look at the next section, which is about instrumentation. No, actually, before we do that, let me just go back to when it talks about spikes, because I just want to say a few things to go with this term. So matrix spike data with the amount of a chemical recovered with a good percentage of about ranging from 70 to 120% means low or no interference. You can find more information about that in a very useful resource, which says called "How to Interpret Laboratory Data: Environmental Cleanup Educational Tool Series" in 2009. Now let's move on. The second sub-move is to describe instrumentation, and it describes the scientific apparatus used in the study. Both custom-built instruments, such as a high vacuum chamber or a newly designed light source, and commercially available ones, like a gas chromatograph or an infrared spectrometer, are described. Ordinary lab equipment, such as a rotary evaporator, is not described. The sub-moves in Move 2 are placed side by side in the move structure in the figure I showed you in the very beginning to indicate that the authors may present the sub-moves in either order to parallel the sequence of events in the study. For example, in Exit A. This is another sub-move, the numerical methods description, but it's not always required. The last move of the methods section is only included if numerical or mathematical procedures were used to analyze, derive, or model data presented in the paper. In such cases, the experimental methods are described, and then the numerical methods are described. Subheadings used to demark move 3 include statistical methods or data analysis. Common statistical methods are described along with the use of subheadings, and when specialized statistical software is used, the name and the version number of the software package is supposed to be reported. But routine software is not supposed to be included. Let's move on to the next slide, where you can see some typical sentences describing, data using statistical analysis. You can see passive voice is also used here. You can see the version and date of the software, and a variety of vocabulary, like something the analysis was performed or the trials was subjected. The mean score was subjected to multivariate analysis, or levels were set. You can find more words, common terms in data analysis methods here, like mean, median, and mode. Log normal variables, which I found in many environmental chemistry papers, convolution integrals, left censoring, coefficient of variation. Now, let's compare these moves and conventions with those of a synthesis paper. Let's have a look at move 2. I want you to pause the video and take as much time as you need to identify differences between excerpt A and B. Excerpt B begins with a subheading with the name of the specific compound that was synthesized. In other synthesis paper, the subheading may refer to a general procedure for synthesizing a class of compounds, for example, tetrazoles. After that, we often see that they go on to describe the steps in the synthesis. Information about specific reagents and materials are included in move 2, where the individual syntheses are described. Did you notice that excerpt B includes the mass, the moles of solid reagents, reaction times, temperatures, description of products like solid white power? Synthesis chemists use analytical information such as melting point and mass spectral information in the procedures. In most synthesis papers, analytical data confirming product purity or composition are reported in the method section, not the results section. What is really interesting to remember, though, is that if you're into organic synthesis, research articles published in Organic Letters, which is another journal, tend to omit a method section entirely. Instead, the procedures are published in the essay section online. Details, as well as what is normally included in their procedures, we also need to see what is normally omitted in the procedures. And you can see here that the equation used to calculate the yield is omitted, a list of disposables are omitted, names of common glassware is omitted, routine procedures such as weighing, diluting, and purifying are omitted, or any mistakes that you might have made are also omitted. Language-related issues in methods section involve the use of common synthesis phrases that they expect you to use, such as the one shown in this slide. When it comes to instrumentation, we need to know two things. That it is very detailed and it's always coupled with a diagram only if it is novel. Novel means new. And it needs to report operating parameters. If it is the first time you report instrumentation because it's novel, you do that in very detail. But the second time, you only need to cite the source, either as a footnote or a reference number or a citation, depending on what formatting the journal wants. Operating parameters are also necessary. They are not instructions of how to use the instrument. Parameters are selected and optimized, and they vary among instruments. They affect the outcome and reproducibility of the experiment. That is why I suggest you read examples of how these things are used and described so that you can actually copy, the style and what is included as important from another published paper in the same field. Now, here you can see a typical general section in the experimental section and the operating parameters mentioned. Organic synthesis papers seldom describe the chemicals in the first part, as we said, right? The first part is used to summarize general reaction conditions. You can see abbreviations here like DMSO, which is the chemical structure in organic chemistry, dimethyl sulfoxide. This excerpt describes compounds and materials and is an excellent place to examine writing conventions regarding capitalization. You probably noticed already that chemists are a bit picky about what is capitalized and what is not. Most novice writers know how to capitalize the following, like molecular formulas, vendors and brand names, absolute configuration RNS, and selective abbreviations like THF, NMR, ACS. I want you now to take your time and identify what is capitalized in this excerpt. Pause the video here. So we do capitalize all these things that I mentioned before. In this table, this is a very useful reference because you can actually notice the differences. The correct versions are on the left-hand side and the incorrect versions are on the right-hand side. And you can notice the differences when it comes to capitalization. I want you to spend some time looking at this because in the next task, I'm going to ask you to identify if there are any capitalization mistakes. So take your time to take notes and see what is right and what is not right when it comes to capitalization based on these examples before we move on. So, my question is, are the following sentences correct or incorrect? Based on your notes and understanding of what the previous table mentioned, are they correct or incorrect? Pause the video here and write your answers. Yes, all sentences are correct. Well done if you have found the right thing. Let's have a look at other issues that are very common in method section. As we said earlier on, passive voice and past tense are very, very typical. As you can see, the mixture was stirred for 10 minutes at room temperature is an example here. Common abbreviations include the following things that you can see here, like DNA and NMR and HPLC and so on. You can see that the compounds are highlighted with bold here. You can also see before that weight per weight, atomic weight, disintegrations per second, and so on. And the last thing that you need to remember is that ordinal language is seldom used. It is not encouraged. So please do not write sentences that start with first, second, third, finally. This is very boring, and usually they ask you to rewrite stuff like that. Let's have a look at what you can do instead. When you describe processes, you can use sequence words like these. So you can use words on the left-hand side in red, like prior to data collection, X was checked, or after data collection, this was done. On completion of data collection, this was checked. Once the data were collected, this was checked or estimated. Following data collection, X was checked or estimated. So you can see there are other options here instead of just saying first, second, third, and so on. Let's have a look at another issue. Most people have issues with units of measurement and verb agreement. I want you to have a look at the sentences and tell me which option is correct. Were you going to say was or were in each of those sentences? Please write your answers now and pause the video. Right, so the first one is to the mixture was added. The second one is to the mixture was added. But the last one we have to the mixture were added. You can see why, obviously, because we have two quantities there. Let's have a look at another issue which concerns joining sentences in passive voice. This is a tricky one, and even native speakers often fall into the trap of making this mistake. The eluent was added to the column and the samples collected in 10 ml increments. When you read this sentence, you might think that there's nothing wrong with it, but actually what is wrong with it is that the passive voice needs to be repeated properly in both parts of the sentence, and the verb to be shouldn't be excluded, especially when the subject, the samples, is different from the first subject, the eluent. Now let's move on and talk about numbers. You can see here an excerpt from an organic synthesis paper and a few sentences from other articles at the bottom. I want you to notice at least 5 conventions, rules about numbers, and write them down before we move on. You can pause the video now. Now, let's just confirm that you noticed some of the following. You can use numerals with units of time and measure. 5 minutes, 40 ml. You can use numerals for numbers greater than 10. 40, 10, 15. But that doesn't mean that you shouldn't be using numbers if it's less than 10. 1 ml. You use words for numbers less than 10 except for units of time or measure, for example, ml. Measure is a general term that implies units of volume, width of length, mass, or temperature. or concentration, for example, M, molarity. Some exceptions include using all numerals in a series or range of values containing the number 10 or greater. For example, you can see in the sentences 5, 8, and 12 samples. So we can use numerals here because they're all in a series. We can also use words for numbers that start a sentence in the very beginning of the sentence, unless the number is part of a chemical name, which is the example shown at the very, very end. Now, tell me if the numbers you see here is correct. What do you think? If you said it is correct, if you said it is incorrect, you're absolutely right. Let's have a look at the next one. Is this correct? Nope. No, not correct either. Let's have a look at another example. Is this correct? No. Incorrect. Now, I want you to write why the second options in each case are, more appropriate or preferable. Pause the video here. Another issue that is very common regarding units of measure and numbers is surnames used with either lowercase or capital letters as capital letters. This is because when surnames are units of measure, then we use lowercase. The word form is also used for numbers to start a sentence, unless the number is part of a chemical name, as we said earlier on. And have a look at the last sentence. And I want you to take some time to think. This sentence is correct as it is, but I want you to take some time to think of how you can improve the last sentence, how it can be much, much better than that. Pause the video now and write your answer. I'll give you feedback as soon as you write your answer with an option that I have in mind. Moving on, we're going to look at more issues that occur or turn up during write-up. We need to avoid single sentences repeatedly, as this may lead to wordiness. And as we've said before in our classes, wordiness is one of the most common issues in chemistry papers because experts expect you to write in a very, very concise manner. Let's have a look at this example. The petri dish was placed on a turntable, the lid was raised slightly, and an inoculating loop was used to transfer culture to the agar surface. The turntable was rotated 90 degrees by hand, the loop was moved lightly back and forth over the agar to spread the culture. The bacteria was then incubated at 37 Celsius degrees for 24 hours. As you can see, there is a lot of repetition, and there is a lot of unnecessary detail, and there are words there that are used that are quite vague when it comes to what really happened. What is not mentioned here is streaking, S-T-R-E-A-K. Streaking is a technique used to isolate a pure strain from a single species of microorganism, often bacteria. Samples can be taken from the resulting colony, and a microbiological culture can be grown on a new plate so that the organism can be identified, studied, or tested. Let's have a look at some better examples. Each table was placed on a turntable and streaked at opposing angles, not back and forth, with fresh E. coli culture, more specific, using inoculating loop. The bacteria were then incubated at this temperature for so long. But this is not enough either. Experts would expect you to omit even more details and write something like this. Each plate was streaked with fresh, overnight E. coli culture and incubated at 37 degrees for 24 hours. So now I hope you can see what we mean when we say that your writing needs to be very, very concise. Concise writing involves eliminating unnecessary words, replacing all the wordy phrases, all the vague phrases with very specific and succinct ones, and using brackets to report information of vendors, reaction conditions, amounts, or product yield. Is this sentence concise? Please take your time to decide what parts of this sentence could go. What shall we ditch? Let's have a look. As you can see, the first sentence, the first phrase, is completely redundant, as we can just add a citation in the end. This is according to the ACS Style Guide. Now, before I close, because we're coming to the end of our video session, I want to let you know that a very, very useful resource when it comes to writing can be found in Academic Phrase Bank from manchester.ac.uk in the section that says Describing Methods, where you can find a series of sentences that other scholars have compiled, collected, gathered together for you regarding describing previous used research methods, giving reasons why a method was adopted or rejected, indicating the use of an established method, or describing a process. For example, describing the process using adverbs and so on. What I want you to realize is that in this academic phrase bank, you can find the phrases there ready, not the ideas, not the content, but the phrase that you need in order to communicate the content that you want to share with others. Now, at this point, I'm going to show you a quiz. I want you to pause the video and take your time to answer the quiz that is in your Google Doc with a yes or a no before I show you the answers below. Let's do this. What should be included in method section? A table of chemicals with their physical properties. The answer is no. Amounts of reagents used in the synthesis? The answer is yes. Directions for preparing a stock solution? No. The quality of chemicals used? Yes. The names and locations of chemical vendors? Yes. Brand names of commercial instrumentation used? Yes. Diagram of a distillation apparatus? Nope. An illustration of a novel or custom-built apparatus? Yes. Operating conditions of a gas chromatograph? No. Equations used to calculate percent yield or dilution ratios? Nope. A list of disposable equipment? Nope. Step-by-step instructions of the procedure? No. And warning to other scientists about unusual hazards? Yes. Quantitative statements of reaction times and temperatures? Yes. Descriptions of the physical appearances of synthesis products? Yes. IR or NMR data confirming product purity? Yes, especially in the QA section. Statistical packages used, including the name of the software? Yes. Reports of other software used to keep track of data, for example, Excel? Nope. At this point, I'm really happy because this is the last slide of our video session, basically summarizing the questions you should be asking yourself when you've actually written your method section and you've come to the end of it. Thank you very much for listening to me and being so patient, putting up with my voice and everything. And I want you to look at these references if you have more time. The first one is the main resource for this presentation, but I've used all these other references in order to put together this video. And before we close, I just want you to remember that for Thursday this week, we have homework, or in other words, a challenge. I want you to make a similar layout skeleton scheme like this one, using examples from an inorganic chemistry paper, an analytical chemistry paper, and an organic chemistry paper. So 3 papers, because the one that you can see here is to do more with what can I say, general common chemistry papers. I want you to do this challenge using 3 papers of your own. Thank you very much for attending. See you next week.